Okay, it's again Thursday, and we're here for the worldwide series on the neurobiology of addiction. And I have my great pleasure to host today, together with Nicola, we're going to host Erin Calipari. And if you can follow us here on live, obviously. There's also going to be a recording. And at the same time, it's also already live on YouTube. But then, of course, you can also watch it at your convenience, particularly for those who live in time zones that are a little bit less accessible to these hours. So now, without any further ado, I'll pass on the mic to Celine to introduce Erin.
Hi, everyone. So it's my great honor to introduce Erin Calipari today for the talk on neurobiology of addiction. So Erin got 2 bachelor degrees in 2009, one in biology and one in psychology at the University of Massachusetts. Then in 2013, she obtained her PhD at Wake Forest University under the supervision of Sarah Jones. There, she studied combining analytic chemistry approaches and operant behavior on how dopamine kinetics are impacted by drug self-administration. In 2014, she joined Eric Nestor's labs as a postdoc at Icahn School of Medicine, Mount Sinai, where she focused on understanding the interaction between neural circuit activity, transcriptional programs, and complex behaviors. Since 2017, Erin joined Vanderbilt University as an assistant professor as part of the Vanderbilt Center of Addiction Research. And today I'm very excited to hear what she's going to present about the latest data from her lab. Erin, I leave you the mic.
Thank you so much. Okay, I'm gonna, every Zoom starts with me being like, can you see my screen? You can see my screen, right?
Yes.
Yeah, we can.
Okay, good. So if someone will have to tell me if there's something wrong. So my career has focused on mostly how drugs of abuse change the dopamine system to alter motivated behaviors. And so, when I started my own lab, we started, and I've been recording dopamine and the striatum for almost a decade now. And so when I started my own lab, we started doing some work where we were recording dopamine during kind of other behavioral tasks. And what I'm going to show you today is going to be less addiction maybe than my normal work, but very relevant for addiction. And so I think one of the things that's really important, and one of the things that my lab focuses on, is understanding how neural circuits kind of what they are encoding so that we understand what these drug-induced deficits actually mean to the individual. And so the kind of overarching kind of goal of my lab is to really understand how neural circuits in the brain are changed by experience. And so how these changes are this kind of circuit dynamics increase the probability of behaviors that result in positive outcomes and decrease the probability of behaviors that result in negative outcomes. And so this kind of fundamental process is really core to almost everything that we do. And so we learn these kind of things very quickly, but what's really important about this process is the kind of balance between seeking out positive rewards and avoiding aversive outcomes is dysregulated in almost every psychiatric disease. And so, in substance use disorder, which is the thing that I study most often and the thing we're all excited about, drug-associated stimuli in the environment are overvalued relative to negative consequences or alternative reinforcers. And so this causes things like compulsive drug seeking. This causes, drug taking in the face of negative consequences. So just understanding this adaptive process is really, really important to understanding how it goes wrong in addiction. Things like depression also show things like reduced motivation, reward valuation, reward learning. Anxiety and stress disorders. Some of the people in my lab are really kind of passionate about things like PTSD, where you have these negative outcomes, but they're overgeneralized to neutral cues and contexts, which causes maladaptive behavior. And so really at the core of all of this is how we learn to navigate our environment and how psychiatric disease changes the way that we do that. And so, the first step is kind of thinking about how we learn to make adaptive decisions. And so we use information in our environment, contextual cues, things about kind of where we are. And so, one example is a war zone versus a hometown. We use discrete cues, things that are directly paired with outcomes, which in the addiction field we talk about all the time. So these are are kind of paraphernalia associated with drug taking, maybe the sound of a helicopter. And so what's really important is these contextual cues can inform what discrete cues mean. And so the sound of a helicopter in a hometown versus a war zone may mean something very different to an individual. And then we also learn about our behavior. And so my background is reinforcement learning, and this is what gets me really excited. And you're going to see kind of an integration of both kind of Pavlovian and reinforcement-based learning to kind of understand how dopamine is driving behavior. And so, to do this, the psychology field's been studying this for years, for many years, hundreds of years. And so, we have to be able to encode the value of an unexpected outcome, like, is it good or bad? We need to make predictions about when that will occur. And so can we predict when something good or bad will occur? And so this kind of associative learning where we associate environmental cues with outcomes is really important for behavior. But what's also important is that we need to be able to update those predictions when they change. And so this kind of adaptive process where we can make associations, update them when they change, is really, really fundamental to behavior in every single context. So there's going to be some math, but don't worry, there's not a test at the end. So if you're not a math person, you just ignore that. There's lots of biology. And so psychologists have been really trying to kind of make theoretical models that predict how this occurs. And so one of the really early kind of models of learning and memory or associative learning is this Rescorla-Wagner or reward prediction error model. And so in this model, essentially what happens is that you have, and reward prediction error models, this Rescorla-Wagner learning model was early. There's been many iterations of this that include things like time and like temporal difference models. But essentially what you have is you have a response to an unexpected outcome. And then as that outcome is predicted and the animal learns the prediction between that outcome, you have a response to the actual predictive cue. And so essentially what they did is they formalized this type of learning. And what it says is that we learn from errors in prediction. So as a cue becomes really predictive and the animal learns that, your ability to predict that outcome gets better and the errors in that prediction go down because the prediction is perfect. And so this has kind of been a really powerful model for explaining how animals learn and a lot of these core associative processes. And what people have been doing for a really long time is trying to figure out, what circuits in the brain actually encode this information. And so where is the neural basis of this kind of associative learning? So, a focus of a lot of this work has been on the dopamine system. And so the dopamine system is probably, everyone has to have their favorite neuromodulator, and dopamine is mine. And so essentially, my career is really focused on studying these dopamine neurons that originate in the ventral tegmental area and project to the ventral striatum. And so everything I'm going to show you today is in the nucleus accumbens core, unless people have questions, and then I'll show you data from other striatal subregions. But essentially, dopamine neurons are critical for survival. Lesioning dopamine neurons prevents reinforcement and Pavlovian learning. And dopamine neurons respond in a fashion that mimics this kind of reward prediction error-based learning. And I'll show you that in a second. Dopamine is critical for addiction because almost every drug of abuse that is reinforcing increases dopamine in the nucleus accumbens. And so DiChiara, showed this with microanalysis for nearly every drug that we study. And so this has really been a focus of both the addiction field, but also the kind of learning and memory and associative learning fields. So this kind of idea of dopamine encoding reward-based predictions and kind of associative, even motivational value comes from this data from Wolfram Schultz and many other people have kind of replicated these patterns where they recorded in the midbrain in VTA. And what they show is that dopamine neurons respond when unexpected rewards are delivered. When a cue predicts this reward, the dopamine neurons fire to the predictive stimulus, they fire to the cue, but they no longer fire to the reward because it's perfectly predicted. So there is no error in that prediction. And then if the reward is omitted, what you see is a firing to this predictive cue and then a reduction in firing at the time of the predicted stimulus. And so essentially the thought here is then dopamine is really encoding these associations. And what it's doing is kind of driving this kind of associative learning. And so This is kind of one of the, and the addiction field kind of thinks of this not always in the same way, but I think for dopamine neurons, especially people who study the VTA, people really talk about it as reward prediction error, prediction encoding, but dopamine does a lot more than that. And again, people who study stress and aversive stimuli know that dopamine is also involved in responses to aversion and motivational responding. It's been linked to things like incentive salience, which is kind of the motivational value of cues. All kinds of different aversive events. And so this kind of, when you're thinking about dopamine, this kind of falls outside of the realm of these kind of prediction-based models. And so we kind of took a step back and said, there's lots of fields that have really done this, but let's kind of look at how dopamine is doing this across these different contexts that aren't often studied together. And so one really important thing is I think the dopamine field has often extrapolated that if the ventral tegmental area cell bodies are doing something, that is what the resulting dopamine release downstream in those same cells should be. And as somebody who's been studying the striatal terminal regulation for a long time, I will tell you that one of the really cool things about dopamine neurons, this is why I study them, is that they are regulated in such a complex fashion. So in addition to VTA firing, dopamine can be evoked directly at the terminals, totally independent of ventral tegmental area cell firing through multiple mechanisms. And the timing, dynamics, the spatial release properties are really shaped by both effectors located in dopamine terminals that respond to dopamine, but also other circuits, peptides, neurotransmitter circuits that are known to regulate dopamine release and timing and how much is there. And so, one of the things, if we want to reconcile all of this data, is we need to directly record dopamine release. And so I'll tell you how we did that in a minute. But the other thing we need to do is a lot of the work people have done has looked at dopamine and motivation. It's looked at dopamine responses to aversive stimulus. It's looked at dopamine responses to rewards. We need a way to understand how to kind of parse these different co-occurring variables that are happening during these behavioral tasks from one another. And so Ganesh Kutlu, who's a research instructor in my lab, and Jennifer Zachary, who's a graduate student, when we first started my lab, they spent a lot of time developing behavioral tasks that allow us to parse different variables that occur in these tasks to try to understand how dopamine maps onto these different kind of factors. And I'll explain to you what I mean. So positive reinforcement is, we all use it, or many of us use it. You see it all over the place. It's really kind of the standard animal lever presses or nose pokes, and they get a sucrose reinforcer, drug reinforcer. This is really standard, right? We know dopamine's involved in that. But what we can do is we can also have these same animals go through negative reinforcement. So negative reinforcement is the animal nose pokes, but they nose poke for a very different reason. They nose poke to remove an aversive stimulus from the environment. And so the reason comparing these two tasks is really important is because they have the exact same action. So if dopamine is encoding the motivational value or the motivation to go press a lever, for example, it will look the same between these two tasks. The outcome value in both of these tasks is positive. So the avoidance of a negative stimulus is positive, and the delivery of a reward or reinforcer is also positive. But the behaviors are maintained by different stimuli with opposing valence. And so what we've done is we've dissociated the stimulus valence from that valence of the outcome. And so now we can start to understand how circuits are involved in positive outcomes, responding to the stimulus themselves, or are just involved in the pure motivation to actually lever press in a situation. So we want to record dopamine directly during these tasks. And so when I started my lab, actually, Lynn Tian was really, really generous and gave me a bunch of D-Light, which is a fluorescent dopamine sensor. And I started using this in the lab. And so essentially what D-Light is, it's a modified D1 dopamine receptor that is fused to a GFP. And so what happens is we express this with a virus in the nucleus accumbens. It's expressed in all the cells. And so this isn't, looking at cell type specific anything. This is just a sensor for dopamine, much like other sensors like voltammetry. It's just optical instead of electrical. And when dopamine is there, the molecule fluoresces. And so what we're able to do now is use different tools. For this study, we used fibrophotometry, and we can now record the dopamine fluctuations on a trial by trial basis that are happening as animals are performing these tasks. So the first data is not surprising at all. It looks like what everyone else found. So first what we did is we had an auditory cue comes on and the animal presses. During that cue, sucrose is delivered. We did imaging. So we recorded dopamine either really early in training before the animals learn the contingencies of the task or after they've met an acquisition criteria. The animals during training, they press more on the active than the inactive, not surprising. They actually are very good at this task. We can vary the difficulty by changing the length of this discriminative cue, which we use for a lot of machine learning analyses. I'm not actually going to get too far into them, but I'm happy to talk about them in the question and answer. And so what we're able to do now is look at how the dopamine patterns change over time. And so what we did is this is the fluorescent intensity, the dopamine signal coming back over time. The heat map is over trials. And what you'll see is early on in training, the biggest dopamine response is when the animals get sucrose. But as the animals learn, the dopamine response shifts back to this predictive discriminative cue. And I'll remind you what this looks a lot like is reward prediction error signaling. You have this increase to the cue, looks like that, and a decrease at the time of the outcomes because a decrease in error. Everything looks great. But then we went into this other behavioral task. So the way the task works is an auditory discriminative cue comes on, and that signals that a series of shocks will be delivered. If the animal presses in the beginning of this cue period, they can avoid all of the shocks. There's a series of 10 shocks. If they start the shocks, they can press anytime during that to terminate them. When the shocks end, either by termination or lever pressing during the cue, this safety cue comes on, which signals the positive outcome. The shocks have been avoided. We did the same thing. We recorded early and late. If you don't know about negative reinforcement, animals actually do this very well. And so we get almost 100% performance near the end where the animals are avoiding all of the shocks. And so we can actually use this to compare. The performance is very similar to the positive reinforcement as well. And so the first thing, which again, if anybody's in the stress field or is a voltammetrist, this will not surprise them at all, as we've been using aversive stimuli to validate our electrode placement forever. But dopamine in the nucleus accumbens is evoked by an aversive foot shock. And so I think you hear a lot of people talk about dopamine as a reward-based signal. I think we're getting more nuanced, but dopamine is also positively responding to aversive stimuli. But what was really interesting is when we started going through the data for this task, I'll remind you again, this is what we'd see if dopamine is a reward encoder or reward prediction error encoder, is that we should see an increase to the discriminative cue and a decrease to the safety cue, which is the positive outcome. But when we looked, we didn't see a huge increase to the negative reinforcement cue, even though the animals were clearly using it to drive their behavior. We didn't put a lot of stock. This is not how we made our conclusion that this doesn't fit. It could be a number of things. But what we did see, which kind of looked okay at first, is that you do see a reduction in the response to the positive outcome or the safety cue over training. But when we look further, the issue with this is that the biggest response to this safety cue was on the first trial before the animals could have possibly learned its value. And so even though this looked like a reward prediction error type signal, it didn't really make sense. And then what we saw is that not only does a foot shock elicit a positive dopamine response, but this actually got bigger as the animals got better at the task. And so the response to the unconditioned stimulus was actually increasing over training. And so what we did is we looked at this data and we said, that doesn't really look like these reward-based predictions, and it doesn't really fit these other motivational models. So basically what we saw is that when we redid the studies that other people have done, we saw the same thing. Dopamine looks just like these reward-based predictions and is associated with kind of motivation to press after a cue. But when we looked at this negative reinforcement, we saw that it just didn't really fit the dopamine patterns we were seeing. So we kind of ran into this problem is that some of the models, not just reward prediction error, but other models too, are a little bit too simplistic to account for dopamine's role in learning memory. And the other problem is what we do a lot is that we have a model and we do experiments to see if dopamine will fit the model, not look at what dopamine does across contexts and then try to see how that could be explained in a theoretical framework. And so what we did is we kind of took a step back and we reverse engineered a model of behavior. And so I'm going to show you this model. And it's a model of behavior, not of the neural signal. And we wanted to understand how dopamine could fit onto the theoretical components of this model. So what we did is we created a complex model of learning and memory. I will not take the credit for this. Ganesh Kulu in my lab is a computational psychologist. This is what he does. He's worked on this for many years. And so what he did is he created a complex model of learning and memory. This model isn't a black box machine learning model. It's based on theoretical studies in the field. And we can break it down into the core components that are actually being able to explain this behavior. He then recorded dopamine in behaving animals across, I'll show you a subset, many behavioral tasks. And then what he was able to do is to actually look at how the different components of the signal we were seeing mapped onto the theoretical components of this model. So I'm going to show you the kind of the core framework of the model, but here's the thing. What we use this model to do is to design biological experiments to test dopamine's role in learning and memory. So after I show you this model, if you want to ignore all the modeling stuff, the experiments actually completely stand alone. They just provide a framework for kind of explaining what we predicted at each step if dopamine was doing one thing. But essentially what we do is we have this model that incorporates these different components. So at its core is actually reward-based prediction still. And so this is really important because if dopamine does map onto reward-based predictions, error-based predictions, it will still map onto this component within our model. However, we also added different components to allow for operant responses and negative reinforcement tasks, things like that. But the most important part of this, and I'll just like give away the punchline, this is what dopamine maps onto, is perceived saliency. So saliency is the physical intensity of a stimulus. So it's things like decibels, lux, amperage, like essentially it's how intense a stimulus is. But the important thing is how intensity is perceived subjectively is different in different contexts. And the reason for that is that perceived saliency is a product of both physical intensity, so how intense a stimulus is, and the novelty in an environment. And what this does is this novelty basically drives attention towards that stimulus. So essentially perceived saliency is a term that explains how novelty and intensity combine to change how we attend to a stimulus. That's what it is. And so using this framework, we went through and said, okay, well, we are going to design experiments to manipulate physical intensity, manipulate novelty independent of physical intensity, and then manipulate perceived saliency in context where error prediction or other terms would not result in the same prediction. The important part of this model is that we can model all of the previous things that reward prediction, temporal difference error models were made to predict associative learning, extinction, overshadowing, blocking. But this attentional term allows us to also manipulate or model things that those models failed at: latent inhibition, sensory preconditioning, and simple operant tasks that are changing and contingency changes over time. And so this is just a couple examples. I can show you many more, where essentially the data from the animals is in blue and our simulations are in gray. Here we trained animals to nose poke for sucrose, and we switched it so that now they're nose poking and a foot shock is given. You can see both the model and the data show punishment behavior. We also showed that the model can predict negative reinforcement, where the animal's nose poking to remove an unconditioned stimulus. So they're responding for the absence of a stimulus, which is actually, really difficult to do. And so this model can do that. So now we went through, we recorded dopamine, where we manipulated different aspects of what drives perceived saliency. So the first thing we did is said, okay, well, if dopamine is responding to just saliency, perceived or otherwise, what should happen is that it should scale with the stimulus intensity of both appetitive and aversive stimuli. And so this is the computation coming back from our model. And so what it shows is that perceived saliency, as the intensity goes up, increases. What we found was that dopamine also does that. And so as we increase the shock intensity, the dopamine response to that foot shock scales with the intensity. And so this is going up for aversive stimuli. But we also did this for sucrose, which is an appetitive stimulus, in quinine, which I think is important because it's an aversive stimulus, but it's a different sensory modality. And so what we found is when we increase the sucrose volume, dopamine is increased. And if we increase the amount of quinine, dopamine is also increased. So essentially what we're showing is that dopamine in the nucleus accumbens core is scaling with the intensity of stimuli, no matter what their valence. And so again, I think other people have also done this, but this rules out simple reward encoding by dopamine release patterns in the nucleus accumbens core. So the next thing we wanted to do is, if it's just intensity, right? It's just salience, then it should only be changing with intensity and not these other factors. But if it's perceived salience, what it should do is it should change with intensity, but also novelty. And so what we did is we did this experiment where we repeatedly gave an animal foot shock, but we kept it at a constant intensity. And what we found was that dopamine goes down with repeated presentations of a stimulus of the exact same intensity. And so repeated presentations of a stimulus, which reduce novelty, reduce dopamine. And we did a bunch of experiments to rule out that this was not depletion. We can rescue this signal by introducing novelty into the context. So this is not just lots of shocks deplete dopamine. It's a novelty effect. And this holds true for not just stimuli with strong emotional valence, but for neutral stimuli as well. So auditory cues that are neutral, that will not go on to acquire value, have no outcome at all, have positive, significant dopamine responses that also go down over time. And so dopamine is responding to any stimulus in the environment that is novel or salient, and it decreases overexposure. So I just showed you the dopamine responds to positive and negative stimuli and also tracks novelty. But we designed an experiment to specifically dissociate dopamine from other terms that has been shown or been suggested to modulate. So what we did, we trained animals on a reinforcement task. An auditory cue is presented. If the animal presses, it gets sucrose, positive reinforcement. They do it great. But what we did then is we explicitly produced an error in a reward-based prediction by introducing a foot shock unexpectedly to the same cue. And so the reason this is important is because when you introduce an error into a reward-based prediction, that is a prediction error. Dopamine should go down if it signals reward prediction error. It should also go down if it signals the kind of motivational value or incentive salience of the cue, because the cue is now represents a worse outcome than the non-shock paired cue. But if it encodes perceived salience, dopamine response should go up to the shock and it should be higher to the cue. And so what we see first is that when we introduce this shock, animals' behavior and the simulations show that the animals reduce their lever pressing, which is not surprising. And again, what we should see to this cue during this switch is an increase in the dopamine response if it encodes perceived saliency or signals perceived saliency. And so what we're looking at here, just to orient you, as we're looking at this cue, so you have the SD that predicts sucrose. The next one before the shock still predicts sucrose. And so we're looking at the cue before the shock and then the next one immediately after the first shock. And so what we see is that the first shock itself, even though it's a worse than expected outcome, it has a positive dopamine response. And we showed that the dopamine response to this discriminative stimulus is actually significantly increased, even though it's an error in prediction, and even though the animal is reducing its behavior. And so it rules out both reward prediction error, because this is an error in prediction. It should be tracking that if it does, but it also just rules out simple increases in dopamine, increasing rates of responding, because this is associated with animals decreasing their rate of responding. So I'm going to show you a few more kind of things and then show you that some optogenetic studies that kind of further parse this. So another thing that's really important is that we're showing that novelty itself can influence conditioned responding and responses. What we did is we trained mice in fear conditioning. A cue predicts a shock. And then what we did is we introduced a novel cue into this task. There is no error in the previous prediction. All there is a novel light. That's it. What the model would predict, perceived saliency would predict, is that this novel light, because it's novel, will increase dopamine responses during that task. And what we see is that the dopamine response, when the light cue is given, only on these light trials, increases the dopamine response, even though there is no error in prediction, and even though that cue does not go on to acquire value itself. And so essentially, it's ruling out simple attribution of valence to environmental stimuli, as we're seeing an increase in dopamine to a cue that does not necessarily go on to acquire value. Another thing that people ask me all the time, if you're really into reward prediction stuff, is whether it could be just valueless prediction error. Dopamine is going up every time there's an error in prediction. It's a great question. And actually, again, we didn't set off on this to prove a theory. All we did is set off on this to say, what is dopamine doing across contexts? And so what we did is we did an experiment where we trained animals in fear conditioning. And then we omitted the shocks, either omitted them or did extinction. And we looked at the time of the foot shock. So what we're looking at here is not the cue, we're looking at the response to the foot shock. Perceived saliency, because it's intensity and novelty, what it should do is when you have a physical stimulus there, both of these are activated, or maybe the intensity is stronger, but you should have the biggest response when the stimulus is present, and a significant but smaller response when the stimulus is predicted but absent. And that is what we see. If this was a prediction error signal, we would expect that the omission response would be bigger than when the stimulus is there. And so what we're seeing is that the dopamine response to omitted foot shocks is also tracking this perceived saliency term. And so this also rules out any prediction errors that do not have a sign on them. So I'm going to show you two more, I promise, and I'll stop. I'm going to show you two studies that are optogenetic studies. So we had done all this work, and some people said, okay, well, you need to show this is causal. And we said, okay, let's figure out using the model how to do this. So if you remember, I just showed you that novel stimuli increase dopamine responses. And when we went back, we said, okay, let's look at the conditioned response to the cue during this time. And what we found is in a phenomenon, this is called external inhibition. So external inhibition is a phenomenon where novelty in the environment reduces the conditioned response to a previously learned contingency, even though there is no error in prediction. And essentially what we found is that dopamine went up, and the dopamine was negatively correlated with freezing behavior during these trials. And so what's really important is that external inhibition is actually a really important kind of problem for other models, because what's happening is you have a change in a response to a learned association, even though there are no errors and there's no change in the motivational value of that particular cue. And so what we wanted to do is to see if increasing dopamine artificially in the absence of novelty could induce external inhibition. So what we did is we expressed channel rhodopsin, light activated ion channel, in dopamine neurons in the ventral tegmental area that project to the nucleus accumbens core. And we implanted a fiber optic over the terminals in the core so that we could evoke dopamine release in those terminals at temporally defined fashion at times. What we did is we stimulated in a trial during there, so 80% of the cue presentations, there was no stimulation. And in 20% of the cue presentations, we optically evoked dopamine release. And what we found, and so I'll remind you, if dopamine is encoding the associative strength of a cue, for example, what would happen is that you should see an increase in freezing because you would increase the dopamine response. But what we actually see is that stimulating dopamine to this cue period reduces freezing, but only on the trials where the cues are the dopamine is stimulated. And so essentially what we're doing is we're inducing an external inhibition selectively on specific trials by increasing dopamine. And what I'll show you too is we simulated the response that would happen if dopamine was artificially increased and it signaled perceived saliency. What we saw is the exact same thing we see here with our optogenetic study, is that the simulated condition response tracks the condition response if dopamine were a perceived saliency and not a prediction-based signal. So the last optogenetic study we did, again, we showed that dopamine is highest when the shock is present during fear conditioning, but is significant, but still lower when it's omitted. So if this is a prediction signal, this is an error signal, like people have suggested, increasing dopamine to mimic this should make animals extinguish faster because that would be helping them learn there was an error. If it's a saliency signal, like we hypothesize, increasing dopamine at the time of the omitted shock should totally prevent extinction of that previous association. And so what we found is when we stimulate, so we stimulated during the shock period, we optically evoked dopamine release during this period to kind of mimic these signals. And what we found is we completely prevent extinction of a fear conditioning association by stimulating dopamine during the time of the predicted shock during these omission trials. And what we found is that, again, when we used our model to predict what would happen if dopamine was perceived saliency, we saw the exact same thing in our condition responses. So I showed you a lot of data, but what I showed you is that dopamine in the nucleus accumbens core maps best onto perceived saliency and not these other motivational or prediction-based terms. Not that it does, they don't go hand in hand. The reason that these look like that is in many cases, salience, perceived salience is going in the same direction as error prediction, as associative strength. It's only when you design experiments to specifically disentangle these do we see that it better tracks perceived salience than it does these other terms. This model that we have is I want to emphasize, is not a neural model. It's a model of behavior. And so one of the nice things is that we can actually map activity onto discrete components for other circuits in the brain and try to figure out kind of what experiments to run to parse those. And again, I think the really important thing here is the fact that dopamine does not look like a value-based prediction signal is really important. And so, I'll end with kind of why should you care as an addiction researcher? You say, okay, that's a lot of psychology. I don't care. I just want to know how drugs change the brain, which is me most of the time. But essentially, why does it matter? So here's why I think it matters. So I'll give you a background on these slides. I always like to highlight the one woman neuroscientist who's made like instrumental or very large contributions to a field. And so I'm going to give you all examples from Sarah Jones, who's chair in the pharmacology department at Wake Forest. So Sarah's career, and when I made this slide, it was 100 papers. It's probably many more now. Sarah's career is focused on understanding how drug exposure changes dopamine in the brain. And what Sarah has shown is that cocaine self-administration, if you look with microdialysis, there are deficits in dopamine release. So dopamine is down at baseline and to a cocaine challenge. If you look at evoked dopamine release with voltammetry, so if you don't know these color plots, green is dopamine. You can see in the control animals, you have normal dopamine responses, but in the self-administration animals, you have blunted baseline dopamine and cocaine effects on the dopamine system. So you have deficits. If you look at ex vivo responses after cocaine, you see blunted dopamine release and changes in transporter function. And this, she hasn't just shown this for cocaine. She's also shown this following other drugs of abuse. One example is ethanol. And the big thing is, well, what do these deficits mean? And what I think is really important about what I showed you is that if dopamine is in the nucleus accumbens, again, I'm not saying dopamine everywhere. Dopamine in the nucleus accumbens core is critical in transmitting salience, which is kind of a detentional term. It's going to change the speed at which animals learn new information across value and context. And so essentially, if dopamine encoded positive valence, like I think we've thought of before, you would want to decrease dopamine to these cues and things to basically reduce drug seeking. But if dopamine encodes something like perceived salience, what you want to do is increase dopamine to these stimuli to help animals adaptively extinguish drug associations, to help them learn when negative outcomes are paired, and to help them learn new adaptive options. And so I think the really important thing here is that understanding what these deficits mean for a circuit and what the circuit is doing at baseline could really kind of inform how we think about treating these patients. So with that, I'll thank my lab. So Ganesh Kutlu is a research instructor in the lab. He's fantastic. He led this project. Jennifer Zachary is a graduate student. She worked on this with him very closely. Stephanie, Shannon, and Maxime were also involved. Maxime, we did a bunch of stuff with kind of deep lab cut machine learning that I didn't get to show that he worked on, and it was really important for the manuscript. Funding, Cody Siciliano, which full disclosure is my husband, so he's required to contribute to my work, and Patrick from his lab, and then Lin Tian, who's been working with us with the optical sensors, and it's been kind of instrumental to this. And I can take any questions people might have.
Okay, thank you so much for a great talk and very enthusiastic presentation. We already have a few questions, so Celine, why don't you ask the first few?
Yeah, thank you so much, Erin, for your talk. So I'm going to start with the first question from Ashraf Mahmoud. So he's asking, how did you make sure that it's related to the first part of your presentation? How did you make sure that the sucrose given creates the equal positivity as the avoidance of the shock? Aren't you still comparing two different positivity?
Yeah, no, this is a great question. And so one of the things I think you have to be really careful about is comparing across reinforcers, like relative value. And I think that for this project, if you look, like the conclusions we're making is from like a holistic kind of picture about the patterns we see in different contexts relative to baselines, omissions, introductions of something new. We don't want to make conclusions about sucrose and shock. I think, for example, foot shock. Foot shock is going to be more salient than most things for a number of reasons. One of which is that the animals can't not attend to it. So for sucrose, if they don't smell it, they don't see it, they may miss it. But if foot shock is applied directly to the animal. So none of the conclusions from this are making conclusions directly about relative reinforcing value or relative intensity of sucrose versus shocks. But I think it is an important thing to think about. And I think as a field, we often ignore that and say, oh, foot shocks here. And I think foot shocks are kind of like the maximal salience situation. I hope that answered your questions. But yeah, I think that they are not necessarily comparable. But the first studies with the reinforcement kind of started us down this path to saying something's off. And then we did more, I think, better controlled studies after that to really parse what is going on.
Thank you. So the following question, where do you think norepinephrine would map into your model given its role in detecting novelty, orienting to silent stimuli and stress responses?
Oh, I think norepinephrine is probably doing something similar. I think a lot of these neuromodulation, like, you know, as I study the brain more, I'm like convinced that the brain is just kind of like generates motor response to salient novel events. And then there's some circuits to do valence. I would be surprised if circuits like norepinephrine weren't doing something similar. I mean, it makes sense for neuromodulatory systems. Like think about dopamine. Dopamine is not driving action potentials in neurons itself. It's acting through G protein coupled receptors, which are filtering glutamatergic inputs from other circuits, especially in the accumbens, that are dictating plasticity in these cells. And so I think that these kinds of salience or attentional signals from these neuromodulators is not really a crazy thought. And so I think that there's probably a large likelihood that it probably maps a lot of the same kinds of factors, which I think is interesting, right? And it's a kind of cool way to think about attention, stress systems, and what they're actually doing to behavior at specific times.
Okay, so now we have a lot of questions. So I'm going to go in the order. So Gibran Koka is asking and saying, great talk, Erin. What would your model predict about something like latent inhibition of a condition reward or condition avoidance like response?
I'm like, Gibran and I are like besties, whether he knows that we're besties or not. I did not plant him here. I will tell you, I have, hold on, Gibran, because I'm just going to pull up something else. I was going to just try, I thought I had these slides, but I didn't. We did that. It's a great question. So actually one of the, do you like that I don't prepare? I'm just going to pull up a figures from something else. Okay. So actually one of the really big challenges to reward prediction models is they cannot account for things that are novelty-based changes in learning. And latent inhibition has been one of the biggest challenges. And so, hold on, I just stopped sharing my screen, I think. There we go. You guys can see that now, right? And so basically what we did is we did a few experiments. I'm gonna like quickly show you. One is that dopamine habituates over time to novel stimuli, which is the first thing with latent inhibition, right? Latent inhibition is novelty-based learning that helps animals ignore irrelevant stimuli in their environment. So first we showed that dopamine tracks habituation to novel stimuli. The next thing we did is we basically show that pre-exposed stimuli form lower associations in the future. And that's because essentially what happens is when they're habituated, they have a harder time learning the association between that habituated stimulus and an outcome. And what we found was that dopamine actually tracks the pre-exposed stimulus. So it tracks that pre-exposure. And then the last thing we did is we did an optogenetic study where we stimulated during the pre-exposure period, and we showed that we can bidirectionally modulate latent inhibition. So we can induce latent inhibition by inhibiting using halorhodopsin during the pre-exposure, and we can enhance latent inhibition, or we can prevent latent inhibition by stimulating dopamine and preventing that habituation during the pre-exposure period. And so this is actually part of the reason that we really do think that dopamine is this kind of this salient signal, this perceived salient signal that's highly influenced by novelty, because it is tracking these factors that can be explained by kind of attentional terms and learning models.
Thank you, Erin. So I looked across the questions. So several questions are relating about male and female, and it's what I wanted to ask you. So Did you use only males? And based on your previous work, you know more better than ever anyone else that dopamine can be modulated differently between male and female, especially in response to cocaine. So did you use male and female, and how your model would predict the modulation of dopamine between both sex?
Right. So this is actually a great question. So we did both males and females, but we didn't for this particular, basically we didn't see any, we saw differences. So I'm going to say there are differences, but we didn't see differences in the patterns. So we didn't see differences in like what signal dopamine is transmitting between the sexes. So for this, we kind of collapsed it and said, this is the signal that's being transmitted. But we do see differences between the sexes in how fast they learn these different stimuli. We see differences in dopamine responses to these stimuli. And actually, one of the projects I'm really excited about is from a postdoc in the lab, Lillian Brady, who's looking at dopamine terminal microcircuit regulation by cholinergic interneurons. And what she's finding is that there's really robust sex differences in alpha 4 beta 2 containing nicotinic receptors on dopamine terminals that changes when dopamine is released and how much. And we think that this is actually a really critical component in gating dopamine release in females to salient stimuli in the environment. And so you'll have to tune in later for the kind of so basically, yes, there are sex differences. I think there are nicotinic dopaminergic interactions, and we will see how those interactions influence kind of these patterns and the development of these signals over time as we kind of do more work.
Great, thank you. Can't wait to see this data. So the next question is from Anna Samar, our previous speaker. So she's saying, beautiful work you've conducted yourself with Sarah Jones shows that the effect of cocaine intake on dopamine function are determined by how the drug is taken. Depending on the pattern of use, cocaine intake can actually enhance dopamine responses to subsequent drug exposure. How does this link to the model you've proposed?
That's a great question. Yeah. So actually one of the really, and Anna has done like amazing work on this as well. And she has a bunch of stuff I've seen, I feel like every day there's like a new great study that she has coming out. So if you're interested in this, you should look up her work. This is such a great question. So one of the really interesting things about kind of dopamine plasticity after drug exposure is the pattern of drug use dictates this kind of change. And so I think, I use an example of deficits, which there are there are lots of people who've shown imaging that show deficits in dopamine, but there's also situations, and probably in the situation, which we just haven't really studied in detail, that show that when drug is taken in an intermittent pattern. And so, typical self-administration models are, what we call long access, where we just give animals like 6-hour access, take as much as you want. Those result in reduced dopamine release. But when we give animals access to drugs on schedules where they cannot just take unlimited drug, that they take less drug, but in kind of these intermittent patterns with timeouts, that we get sensitized dopamine release. And so I think what's really important is that this might mean that we need something like personalized medicine, right? There might be individuals where you do have increased dopamine responses, and that is part of the process that helps them learn associations between drugs faster. But then there might be individuals that have these massive deficits after really long-term use. And this might vary from drug to drug, right? It might be something where with cocaine, we kind of see maybe more sensitization, but with other drugs like alcohol, where you see really large binge drinking and things like that, we see deficits. And so I think this is something that we really need to start thinking about and looking at in the human population. And there just have not been great studies looking at pattern of intake and how people take drug and how that influences kind of neuroimaging endpoints. But people are starting to talk about it more. And I think that will be something people start to look at in the future.
Thank you. So the next question is from Munir Sevik. And how did the pattern of dopamine increase change during 10 versus 30 seconds conditioned stimulus? And did you see Weber-like constancies?
Yeah. So basically, actually, I'm gonna answer a question that's kind of your question, but is another question as well. Let me find this data. So one of the things that we started doing was changing the cue timing. And so the reason we did it on that task was to make it more difficult for the animals, so that we can have dynamic range for kind of machine learning. But we did another task where we changed cue contingencies. And this is actually a really important point. Let me see if I have, of course I have like a disorganized slides. There's somewhere. Okay. So I'm going to kind of go a different direction. So one of the things that people asked us, which is a really good question, by the way, they said, okay, other people have done voltammetry during fear conditioning, and they see reductions in dopamine responses to these cues. And we said, so do we. And they said, well, isn't that the negative value? And we started looking and we said, we don't really think so. It doesn't track the behavioral responses we see. So we designed an experiment that that basically modulates the action of the animal at a specific time. And so what we did is we basically trained animals to nose poke for sucrose, positive reinforcement. We saw this really big increase in dopamine response to the cue. And then what we did is we changed the task to what's called negative punishment. Negative punishment means if the animal presses during the cue, the reward is removed. So what the animal has to do when the cue goes on is they just wait. And they just wait for the reward to be delivered. The important thing here is both cues are equally predictive of sucrose delivery. Both cues result in the same number of sucrose deliveries between animals. But what we saw is that even though both cues predict sucrose reliably, that the cue where the animal had to actually wait reduced dopamine release, and the cue where the animal had to motivate behavior towards the cue increased dopamine release. And so what we're seeing is that these cue timing and task parameters that require different kinds of attention, they're tracking things not like valence, they're tracking perceived saliency, which is an attentional term. So when the animal has to just wait, attention is low because they're just waiting and it goes down. When the animal has to motivate a response and exploration is adaptive, dopamine goes up. And so basically the dopamine response if you're just looking during the positive task, if the cues get faster, the animals just get better at the task and the responses. We haven't compared like whether the response is slightly higher. That's actually a great question, but it was positive regardless. But when the cues are different, so the contingencies change, we can change the cue response, even though the outcome is the same.
Thank you. I think we have time for one or two more questions from
Sure.
two more. Elizabeth Donchek. So great talk, and she had three questions. You already answered to one. So the first one is, what would you predict you'd see if you recorded dopamine responses after switching from sucrose to quinine rather than food shock?
Yeah, I think that's a good question. We haven't done that yet. We've talked about doing it. We started with sucrose shock because the thing with shock is that it gets delivered and the animals all experience it. The problem with quinine is that they have to actually go in the port and they have to drink it. And we wanted to start with a kind of just like, let's do something that will work and the animals will perceive it. We're starting to go back and do some of this stuff with quinine, but what we're doing is trying to think about how to do intraoral gavages. In mice, it's quite hard. But what it does is it allows you to non-contingently deliver these stimuli really quickly so that you don't have the confound of whether animals actually consumed it or not, or whether they approached it or different timing. And so that's actually was a really important part of our task, but I don't know. I assume it would be similar. It might not be as robust because it's not going to be as aversive to the animal. But I think it's a good thing to do. And we've been talking about ways to do it. And it's just easier said than done.
The other question is it still possible to review dopamine signals that may have responded to other food shock provoked behaviors, such as defensive darting?
We did. Yeah, we can definitely do that. We haven't, actually food shock stuff is really interesting. My husband and I have been talking about this a lot. The freezing is not the unconditioned response to the cue. It's actually darting, and the freezing response actually develops over time. And so, it's we've been talking about going back and looking at some of these behaviors. We haven't done it necessarily for the foot shock, but we did do, sorry, just stop my screen sharing. We did do it for a lot of different other behaviors and map dopamine onto kind of attention. And so we did that, but that's something we're going to go back. We just had some machine learning stuff set up, and other people in my lab are trying to figure out kind of more nuanced behaviors rather than these kind of global changes. But I don't have a great answer for you yet. At least I don't have all the answers. That means I will have a career where I can keep doing these things.
Yeah, definitely. So the question I would have had for you is about the cell type diversity within the VTA. So you alluded to some of the work of that, yeah. And obviously you focused on the core of the accumbens, so that limits to some of the cells to project. But is it really totally different in the shell? And
no.
How do you,
no.
So how do you think this specificity comes about?
Let me share my screen again. Yeah, I think it's really, so here's the thing about dopamine is that I think that there probably is a lot of cell specificity, but and I should bring this picture. I don't know why I don't. Dopamine neurons are so cool. When you do cell filling in the VTA and you look at their arborization, they cover almost like the whole, like one single cell will cover this wide area of the striatum. So it doesn't seem to me like this VTA firing just causes action potentials in every single terminal and dendrite. And actually, David Sulzer has done a lot of work looking at calcium relationships of calcium influx, and it's not one-to-one with dopamine release. So there's multiple factors that converge. And I think what you have in an area is the situation where you have an action potential. It's like almost like a coincidence detector, right? If you have an action potential and the terminal, like you have maybe cholinergic regulation coming in through nicotinics and some of these other factors, it primes certain subdomains for release at certain times. And so the signal that you're seeing is just kind of an integration of all of these inputs all at once. I do think that there is some subregion specificity in dopamine release, but I think if you're looking at dopamine release rather than VTA, I think it's maybe less yes, no, as like a cell would be. We did this study again in the shell, the fear conditioning study. What's really kind of interesting is we still see a response in the shell to an aversive foot shock. We see the same kind of reduction in dopamine response to the cue that develops over time, but we didn't see as robust of a scaling with intensity. So it's possible that there are different kind of components that are reflected in these dopamine patterns. And maybe, I mean, we're starting to do some stuff now. Cody Siciliano next door to me does 2-photon. And so we're starting to look at kind of micro domains of dopamine release with 2-photon imaging to be able to see if maybe there's like different patterns that aren't resolved with something like fibrophotometry. But I think it's a complicated question that we're just now starting to get the tools to really parse the relationships with.
Okay, so thanks. With that, we're coming to the end. Again, thank you so much. And I really like the 20 minutes of discussion, very lively, lots of questions. That's how it should be. So let's close here. We're almost, we're exactly at 1 hour. And please come back next Thursday. Mary Kay Lobo is going to be our speaker, and we're really looking forward to that as well. Okay, have a nice day, evening, whatever, and take care.
Thanks, everybody.
Thanks.
See ya.
Bye.
Okay, good. They go well.
Yeah, very good.
Okay, perfect.